Good morning, everybody. Participants online, can you hear us okay? Yes. Great. Okay, wonderful. If everybody could please type questions into chat, we would appreciate that. Thank you.
All right, I think we'll get started. I mean, it's really a great pleasure today to have Mike H, or I can try to attempt to pronounce his name, Mike Hawrylycz. Did I get it?
Yeah, Hawrylycz.
Okay, all right. Anyway, but he gave me permission to call him Mike H. Many people refer to him that, from Allen Institute, to give us this 2-day tutorial. I think it's really, so Mike actually has a very interesting journey. He's trained as an applied mathematician from MIT, his PhD in math. And then he spent a few years in Los Alamos National Lab. But for the last more than 20+ years now, Mike has been devoted to try to use some mathematical and computational tools to analyze different type of data, in particular in recent years, focused pretty much on the neuroscience data. So he can tell you more about what he has done, but today he's going to give us an introduction, especially to people who don't have the background, computer scientists who wanted to understand the data and also to see what are the challenges and issues involved. So I really very much appreciate him coming to give us this tutorial. Thank you, Mike, and I'll let you start. Yeah. Welcome.
Thank you, Yusu. I'm grateful that you provided this opportunity to allow me to speak to this nice program and people. So I have a lot of material. And I sort of debated and thought about, what kind of approach to take, with this kind of sort of series of kind of lectures material. And, one could do a couple different things. I could say, knowing that people are, let's say the audience is largely computational in nature, one could go into a deep dive of machinery and very specific things, but, that also it has some disadvantages. One, it tends to be very specialized to one or two different things. Two, it may become fatiguing after 4 hours. And so I'm going to talk about a lot of material that neuroscientists would talk about and be interested in from a computational perspective. And we will increase as we go, we'll increase the quantitative focus, okay? But in the beginning, we're going to be pretty high level, surveying different kinds of data types, problems, things like that. I'll always talk from a quantitative perspective because I can't talk any other way. Really. I'm not a card-carrying neuroscientist, okay? So I'll talk from that perspective. But don't worry about it if you don't, we're not going to always go into specific algorithms for doing things. I don't know that that's really most beneficial to people, really. I think it's better to get the higher level picture of because you learn all kinds of techniques. And the question becomes, how could I apply these techniques to these kind of problems and understand these kind of data? So that'll be a bit of our focus. So in fact, there are a lot of parts here. We won't finish them all undoubtedly in 4 hours, okay? So, but you'll have the materials. I gave a bunch of papers that to Yusu that I think she maybe makes available to people. And there's actually 8 parts, but we won't get through all 8 parts probably, and but you can always write me if you're interested in some of the things, and we'll talk about it. So this first part, so let's just review the parts. Here are the parts here. It's the first one is cell types of the brain and their characterization. What do we mean by cell types? What are the attributes that we measure? What characterizes them? How do we characterize them, right? Part 2, and then we're going to we're increasing kind of focus as we go through. Part 2, we'll take a deeper look at cell morphology, shape and structure, and connectomics, how neurons connect to each other, how cells connect. In 3, we'll talk about network approaches to cell type data. In 4, we'll do kind of a deeper dive on the analysis of transcriptomic data. In part 5, we'll talk about making connections between data types, multimodal data sets and types. In part 6, we'll talk about strategies for integrating across data sets. And finally, we'll talk about mapping. We probably won't get to part 7, okay, but maybe let's pretend we will. And we can always talk about it. And we can always skip over things, okay? If something is not so, you know, I have a lot of material and a lot of the slides, I'll probably just go through quickly through some things. I'm not going to dwell on slides for a long period of time. So what are our objectives here? Here's some learning objectives, okay? Identify characteristics and significance of cell type data in the brain. Okay? Learn about primary data sources and how to use them. What's there? Understand major analysis techniques and strategies. Apply techniques of multimodal data integration. Right, you will know a lot of techniques. My feeling is that you have learned a lot of in data science. You will have learned a lot of approaches, and when I show you the connection, I think you'll see, oh, I could apply such and such here, right? And we can talk about that too. You can always, if you always have an idea, just interrupt me or raise your hand or something. Okay, you people here get special privileges because you showed up. Okay, all right. So okay. So, and then we'll talk about certain machine learning techniques in slides are all available. In fact, every time, maybe what we'll do is that when we finish a segment, we'll post the slides. Yeah, so yeah. Yeah. Right. I only take emails. I'm not taking questions from you guys. No, just kidding. Okay. And finally, we can talk about projects for further analysis and things that you may wish to do or things like that. Okay, so let's just dig in here. So there was people have been interested in cell types for a long time. It goes back to the original work of the great neuroscientists like Ramon y Cajal, who undoubtedly you're familiar with. He's sort of like the Ramon y Cajal, Santiago Ramon y Cajal was a Catalan. And, he was from Catalonia. He was very, he's sort of regarded as like kind of one of the grand kind of fathers of neuroscience, right? Neuroscientists kind of, they kind of revere him as if you're in a neuroscience program, you will know that they revere him beyond all imagining. It would be like as if every time you gave a talk, you had to mention Carl Friedrich Gauss, okay? Every talk, you had to have Gauss's name on there. It would be something like that, okay? Yeah. So, but anyway, neuroscience had been interested in cell types for quite some time, right, of course. But a big initiative started in 2013 under President Obama that was called the Brain Initiative. And this idea was to try to get bring technologies to bear, new technology, the realization that technologies really drove biological progress, and to get improved technologies for the profiling of data in the brain, right, of cell types of the brain, of functional aspects of the brain. And this was their charge here, exploring the patterns of activity, spatially and temporally. We can understand circuits, get a comprehensive understanding. This was a charge that the White House made to a certain working group to do this. And they sort of set up a starting point. They wrote this report called Brain 2025. It was written in 2014. You can find this report online. It's worth taking a look at. It sets out the scientific vision for how the people would go about understanding and profiling cell types of the brain. And there's a quote from here which I just love. If you're a mathematical person, this is kind of a great quote. It says in there, the brain contains many classes of neurons and glia, but not infinitely many. Well, that's sort of hilarious to a math person. How could there be an infinite number? You're a finite creature. You can't only have a finite, right? But what's the spirit behind that, though, is that it is a very fundamental question in neuroscience. Is to what extent are there cell types, right? To what extent, or do things fall into classes? Do they actually, there's more continuous transitions? Does transcriptomics actually differentiate between cell types in a really meaningful way? And you might say, well, all we have to do is pile all this data together, this gene expression data, electrophysiological data, morphological data, et cetera, and we'll cluster it and we'll see some types will come out, right? Well, unfortunately, it's not really that simple, as you probably know. And one of the reasons is just that there's tremendous subtlety and a lot of kind of continuous sort of gradients in a lot of these patterns, right? Even classes of cells and cell types do not differentiate as easily as you would like. And furthermore, the association between these modalities that we'll see are you know, kind of are as problematic. So we bring in, you know, the major toolkit that neuroscientists would bring to studying all this would be, well, we have our transcriptomic data here, right, is a big thing. And that's only more recently with the advent of more high throughput techniques in molecular biology, right? You know, 20 years ago, we had things like northern blots and other kinds of things, and, you know, electrophoresis and ways that really much more primitive ways of determining whether a gene was active. Then we went through a phase of being able to, you know, microarrays and all kinds of things. And ultimately now we can profile, you know, we can pretty reasonably accurately profile the gene expression in a single nucleus of a cell, right? And we're going to talk a lot more detail about that, okay? But this is just kind of the high level. There's physiological, the firing patterns, right? And firing patterns are the only thing amongst these major kind of attributes, which are really what you would call functional, right? It actually is actually, you know, very connected with the function of the cell, not its genetic origin or you know, its anatomy or shape or structure. But one of the troubles with it is it's a very, I mean, there are unequivocally different classes of electrophysiological cells, but there's also a lot of variability, okay? Even cells of a given type across transchromic will exhibit different firing patterns from bursting to fast spiking, et cetera. And so this is a little bit, you know, and it's also a little bit different. It's more difficult to actually measure in a robust way because the output is conditioned on a stimulus, right? You have to stimulate the cell in a certain way, and that's a parameter, right? That parameter, whether it's an initial thing or it's a series of things, and you know, because we know that neurons will potentially collect input, you know, from up to 10,000 or more kind of synaptic connections before it really decides that it's going to fire an action potential, right? So this is complicated, but you know, we combine these approaches, the morphology, the physiology, the transcriptomics, and to try to obtain some kind of data-driven classification, right? And that's really, in some sense, that's the best approach that we know of. So you know, neuroscience has been, in a way, we find ourselves in a difficult spot where a fair amount is known about the brain in a really micro way, right? We know a lot. We know a lot about ion channel function. We know a lot about, you know, kind of action potentials. We know a lot about computational modeling has explained a lot about cell type, function, and variability. We also know a lot at the macro level, right? In fact, almost everything we know about quantitatively we know about brain behavior has been done through fMRI, other kinds of things, right? We do experiments. We say, oh, this part of the brain is active in certain kind of things. And, you know, local modelers sometimes have a tendency to kind of underestimate that, but it's actually, you know, there's a lot of value there, in fact, and it's actually very important for the high level architecture of the brain. And what's missing, though, is that what we call mesoscale kind of a place, the place that connects the macro large-scale functional areas with local function of neurons and glia, okay? And that comes, to understand that, we need to understand the circuits, right? The circuits, how neurons are built into circuits, what is the role of circuits in the brain, what is circuit function? And we can do that, we can study some of those using genetic tools. We can do kind of a stimulus. We can do multi-patch, record from multiple kind of neurons at the same time, which we'll talk about more about that kind of stuff. And an attempt to, you know, understand these circuits. You know, there's been a lot of theories about circuits and how they're structured in the cortex, for example. You know, whether different areas of the cortex have reproducible, repurposable circuitry, right? And we don't really know, you know, we have some insight into this may be happening. We know quite a bit about layer communication between things, and it appears that that occurs kind of across areas, but we don't have it. It would be great if we understood the computational circuit function more clearly, you know, that might advance AI, in fact, you know. So cells and cell types, there's a bunch of points here regarding that. You know, we don't don't have to go through all these, you know, to read them all, but you know, why the cell, why cell type characterization, and that, you know, it's because the cell is this fundamental processing unit, right? We know that cells, they matter. It's the really the smallest completely functional unit in an organism. It contains everything, power stations, you know, right, ability to kind of reproduce, et cetera, control of its local environment, et cetera. So understanding the genetics of this, understanding the function of this is something we would really like to do. And now with modern techniques and tools, we can kind of do this in a kind of an unprecedented way, and we can build standardized data sets, you know, to do so. So, but cells are heterogeneous, you know, the things that there's a lot of variability. What we would like to do is build comprehensive, unbiased, quantitative, standard kind of models of this, and then perform data-driven kind of computational approaches to these, right? So again, like I said, I'm going to keep. I just prepared a lot of material. You can don't feel like that. You know, if I'm going faster things, this material is all available to you. You can read this. Think of this as homework. You can study it. I just want to rate. I'm just going to address the most important points that I feel are important in this, right? So, you know, this goes back. So we were talking about the great Ramon y Cajal, you know, who actually sat there, and these were the first things that when we started to understand the cells of the brain that we did. And probably if you're in neuroscience, you will know of the famous kind of intellectual battles between Camilo Golgi and Santiago Ramon y Cajal. You know, that basically they, you know, it was Golgi who was more of the technician and who developed some of the techniques for actually seeing and visualizing the neurons. But he believed that there was, in the so-called reticular theory, that basically it was one big web. Everything, it was a network, essentially, a giant network. And Cajal, through his perseverance and actually scientific insight as well as artistic ability, went and really examined using these stained kind of sections. He drew these kind of patterns, these remarkable patterns that, I mean, even today, if someone were to draw this, you would think, wow, that's really cool. You can actually buy artwork on this, and you can buy artwork of this on kind of steel plates and stuff. It's really cool, yeah. But he drew all these, and he understood a little bit, and actually another individual, this Szent Golhai, a Hungarian, was very involved in understanding some of the kind of canonical connectivity motifs they saw. All this was done through what you would call histological approaches, namely you stain a data, or cytological. You stain the data, either you stain all neurons, you stain some neurons, you stain tissue, and you look at it. And you look at it through the microscope, and you draw what you see, okay? And you see these patterns, and you see, evidently, there are different sort of cell types, right? So, you know, this kind of thing, somewhat before really, so you know, in 2008, let's say, certainly we had, well, this was an era where when we profiled gene expression in 2008, it was largely done using, the most sophisticated thing we had were largely microarrays. Maybe you know about microarrays, right? Microarrays were, I don't want to really go into what they are too much, but it was basically, you know, essentially, you couldn't determine everything that was expressed, but if you had sets of sequences of things you thought were important, you could build these little kind of oligos and plaster them on this little array, and then you could take your array and put tissue on that and see what lit up. Okay, that was kind of the way it worked. And so this was about, it was only, by today's standards, marginally quantitative, right? You could get a sense of more expressed, less expressed, but there was no digital readout, okay? It was not something you could do real major data analysis on in some way. But there was a group that met, actually, they met in the hometown of Ramon y Cajal. There's some colleagues that I know very well. Rafael Yusta, professor at Columbia, and Giorgio Ascoli from George Mason. Maybe you know some of these people that they had a meeting, and this meeting was held in this Cajal's hometown, in which they tried to understand they tried to outline the features that mattered for characterization of cell types. And they wrote this paper. You can look it up. I don't remember what it's called. It was called the PETIA Convention, P-E-T-I-L-L-A. And what they do is that in this meeting, they discussed, and I'm sure they had good dinners and stuff too. They didn't work all the time, but they came up with this list of these things that were important in cell type classification. And this paper is available. This paper may be in your set of your recommended readings. I think it is. But they sort of identified all these different things here from, what mattered molecularly were transcription factors, neuropeptides, structural proteins, et cetera. Then there were physiological patterns, the kinds of firing patterns that matter. You can see down here, back propagation, action potential measurements, et cetera, right? And morphological features of shape, orientation, things, right? So these are, they outlined all this. And then they, for example, in terms of the electrophysiology, they characterized the kind of cells that they observed and saw, and from bursting, continuous firing patterns, and a lot of, for example, a lot of computational neuroscience is really devoted to the physiological analysis of data, right? What do you do in when classical computational neuroscience has overwhelmingly been devoted to modeling, neuronal modeling, right? You build models, either abstract models or more biophysically realistic models of neurons. How do you do? What's the abstract way? Is it think of more of a conventional neural net, right? You build a model, things come in, but you have some more, you have more sophisticated dynamical model about when a threshold will be realized and when something will fire. But you don't worry too much about the structure of the neuron, right? But with the increase in our ability to really use big computing resources, people have been doing a lot more what we call biophysically realistic modeling. And that's where we actually we use things like from partial differential equations, we use cable theory and things like that to basically model the compartments, the propagation of diffusion in compartments, and we're able to build more realistic models, right? And we study these kinds of things here. We study how they model. And now, of course, this is a really micro analysis. It doesn't really give us a big picture, but it's important for understanding the dynamics. So there's a lot of approaches here to this cell type characterization problem here. It's just kind of some of the things that are outlined here. And today, we have many, many techniques. We have techniques that we have high throughput transcriptomic things. We have epigenomic techniques that we'll talk about in a little bit. We have morphology. We can now, one problem with the shape and getting the shape of capturing them is that the classical way we do it is we take a slice of tissue in the brain and then we stain it using say something biocytin or something like this. And then we can use some kind of machine learning algorithm to attempt to segment that, right? And so we see the structure of it. The trouble is that it's confined to the local plane, which may be 25 microns thick, 50 microns thick, not much more, right? And so what you get the dendritic process of the neuron very well, but you don't get its axonal connection. You don't know where it projects to, okay? And so there's techniques, what we call full morphology, right? And we're going to talk more about these in detail later for getting the entire axonal process, right? We often use genetic manipulations in cell type classification. Here, so what does that mean? Well, it means that we build transgenic mice, right? And these are mice that we know will express certain genes, or if they express certain genes, we know that, for example, we can make them light up upon imaging, right? And the advantage to that is that, so the unfortunate thing, we don't really have an ability to target very many cell types this way, a considerable number, but not anywhere near the number that we have identified. But we can think of it as a little bit of a Bayesian approach, namely that we're going to condition on having identified the transgenic mouse that's expressing a certain thing, right? So if we know that if we condition on this this transgenic mouse that say expresses certain inhibitory cells, then we can narrow down from there by further analysis what else it expresses or what other type it is. So it's kind of a conditioning tool, yeah. And there's all kinds of other techniques, connectomics that we'll talk about later too. This is just a thing. There are a lot of ways of molecular kind of characterization, a lot of techniques that matter. And we're going to talk in detail in part 3 about how we do those kinds of things. In terms of the cell's function, there are at least two types of gene classes that really matter, right? One are the transcription factors. These are things that guide, determine the kind of the molecular program of the gene. What's happening? When will it be active? When will it shut down, etc. And then the other are, for example, neuropeptides, which exhibit another kind of more diffusive communication between cells. And we'll learn a little bit more about those too. But as I mentioned, there are a lot of confounding elements here. For example, this apparent continuity or gradient effect in gene expression that we don't see always distinctive classes. Then there are considerations of really interesting in development, right? How things change in development, and you may need to consider the developmental trajectory of an organism to fully understand its kind of terminal state. You've probably seen that classical diagram of Waddington that shows the flow of kind of he shows that starting from a given place how certain events take place, and it divides to specializing to a terminal cell type. So, and there, like I said, like I was saying, there are these dynamic changes in cells and states. There's a question of one thing that is always a confounding element is it a cell type or is it a cell state, right? What does that mean? And what does that even mean? Cell state could mean a cell could be doing different things at different times, right? And so we try to understand whether this is something that is, is it part of a circadian cycle? Is it a part of a metabolic state? Is it only responding this way due to certain activations or behavior? Or is it something that's sort of, an unequivocal type that we can reproduce and get this kind of thing? In terms of we're going to talk about how we group and we're going to talk about how we, in a sense, form the types in all of these modalities. The transcriptomic type, let's say first, which is the approach which is also in epigenetic data, which we'll talk about, and I'll define that a little bit better, is it's often done in a hierarchical fashion. And so that's not an obvious thing or a trivial thing, but it's important to understand why you might want to do that. Like, for example, we look at the top part of this diagram here. This is kind of a characterization of neurons, right? We have neurons, we have neurons, we have non-neuronal types, okay? The glia, the oligodendrocytes, immune cells, vascular cells. These are all cells, epithelial cells. These are all cells that also exist in the brain, and they do important things. And some people feel that, neuroscience is overwhelmingly biased toward neurons, right? And well, why? Because what matters, we think using the processes of neurons. But it's become clearer and clearer that many disease states in the brain are really related to glial and other non-neuronal functions. And furthermore, that these glial cells perform a very important role in, maintaining the structure, as we know, even your basic neuroscience, you learn about how sort of myelination takes place, and its glial cells are responsible for, determining this wrapping of neurons for their long projections, right? So the oligodendrocytes, but, so there's a lot of symbiotic activity there, and you couldn't have one without the other. You could build a computer probably with only using insights from the neurons, but you're not going to build a living organism functioning in a hostile world with only neurons. Okay. So we separate these groups, you know, and then there's neurons that are from the central nervous system versus the peripheral nervous system, right? Things that the way motor cells fire is remarkable, like the projection to the extremities of in you know, long extremities in your body, and what happens, how these are activated. They act in very stereotyped ways, sometimes clearer ways than what happens in the central nervous system. You know, and then we'll mostly focus on CNS neurons here, though, you know, and these separate, you know, we separate them into two major types, right? One are the you know, inhibitory neurons and the excitatory neurons. Now, you know, you probably, suppose you wanted to be, if you wanted to, and we now know by doing all this classification, we know that we can identify a lot of types. And we'll see when we look at this data, some of the big transcriptomic data sets later, that, you know, we now can identify reproducibly thousands of different patterns, some transcriptomic patterns in the brain wide, okay? But we don't have any idea what most of them do. And you probably don't need them all to build a super intelligent thing, right? But they for some reason, maybe you do need them to build a living biological organism that does what we do. We don't know, okay? But one thing is clear. You need excitatory neurons and you need inhibitory neurons, right? You can't, the excitatory neurons essentially are a go forward kind of projecting thing. They say, you know, if this happens, then take an action, right? When they collect information, it's a positive reinforcement feedback loop, right? And the inhibitory neurons are more the kind of sort of mediating, let's slow this process down, let's have some kind of negative feedback. You know, without inhibitory neurons, you have a epileptic state. You don't, that's what happens in epilepsy, is a dysfunction partially of this, you know, we don't know exactly if it's inhibitory, but it's a dysfunction of this circuitry, you know. And so you need at least those types to build a functional circuit. And we also know, what some of the major types do, like parvalbumin expressing, vasointestinal protein expressing, other ones, we have functional ideas of what some of these do. But below that, we don't know so much. You know, we'll talk about a little bit about that. But so, and why the hierarchical organization, though? And so the reason of that really is because it turns out you can't really perform, because of this idea of neuronal versus non-neuronal, inhibitory versus excitatory. There's a natural multi-resolutional kind of organization. And you don't, for example, if you were to attempt to see through some massive data splat and unbiased clustering to observe these local types, it would all be obscured in the noise, or overriding noise of longer, higher order effects. I mean, for example, you couldn't distinguish between certain subclasses of inhibitory neurons if you had all the excitatory data in there too. It's just too messy, okay? And so we use typically these hierarchical approaches to narrow things down, right? That has its own potential problems where you might induce bias that way. So you got to be a little careful with that, but it's a general strategy. Another technique that was used a lot is so we talked about connecting these data types, right? We want to understand, you know, if we're going to get the whole picture of a classification, we have to understand how these different types kind of link. And one of the major problems was always that, well, we could record from cells. These techniques have been around for a long time. You patch a cell with a small microscopic pipette, you inject a current, you can record from it. If you're in a neuroscience program, you probably did this, right? And you can measure function of the cell, right? You can also stain the cell and reconstruct its morphology, and you can actually take out, even in a single cell now, we can take out its RNA and profiling it. But part of the problem is connecting these modalities. If these are not recorded in the same cell, then it's really difficult to actually associate these patterns. And so this was a technique that it was kind of pioneered by a group, several people involved, but someone who really stands out is Andreas Tolius from Baylor, who in his lab did a lot to extend this patch-seq to multimodal. And now we can actually record, the technique works that you basically, you patch the cell, you functionally record from it, then you actually use that same pipette to extract the cell's genetic, RNA, which you ship off to sequencing. And then you fill the cell, destroy the thing and fill it so that you can do the morphological reconstruction. Now, it all sounds great. It'd be great if we could do this, time and time again. There's several complications. One is that, as you can imagine, getting all these three to work reproducibly each time introduces a lot of error. It's a lot of destructive. It doesn't always work. It's slow too, right? You can get, we do this at the Allen Institute. We do this. We have thousands of cells profiled in this way. And it's not even necessary, let's say, to do this for every cell, but we do it as kind of linkages. Once we know something that we want, or some in that space, we can use this technique to attempt to build landmarks as to how to link different things. For example, when you see this pattern, we know in the same cell, using the patch-seq technique, we can link it to these other cells. Okay, so it provides a kind of a bridge. I mean, ideally, you would like to do this all the time, but it's time consuming, it's a little expensive, and it's also noisy. There's a failure rate. Okay, the failure rate is pretty high, right? Yeah, maybe 30%, maybe at least, even if you're good, right? Okay, so and I'm not going to dwell on this. There's more information about that in these slides. How we go about combining that data. We don't need to talk about that now. Recently, as I said, this brain initiative it had a group we called BICCN, the Brain Initiative Cell Census Network. And we were involved in that. We have a website. If you go to biccn.org, you can see a description of all this. You can see this. And we wrote a paper summarizing all. There's a paper, which I think that's in your materials, about all the data and its organization from this. So I don't need to tell you too much about that, but it applied all these techniques. And this was done initially in the first round, this so-called Brain Initiative Cell Census Network, mostly to the mouse. It was mostly done in the mouse. The idea was to really come up with the grand solution in the mouse. And we also though did prototypes in human data too. Okay. Now we're into the more human and non-human primate phase, and this BICCN is officially wrapped up. Right. You have to declare victory at some point, even though you haven't really solved the problem. Have we characterized all the cell types in the mouse brain rock solid? No. We have a template, right? Just like the Human Genome Project, right? When it first finished, the draft sequence completed like in 2000 wasn't even really, it was really drafty, and it overtook many years even to get a really more complete version, right? For a variety of reasons. Some of the areas are hard to sequence. Some things don't work, too well like this. This BICCN first focused on this area of primary motor cortex. Why did we focus on that? Well, we focused on that because it was an area which had a very similar structure in mouse, in marmoset, a non-human primate, and human. And so we could actually apply there's a collection of papers that were published in October 21, which summarizes all the work. It was basically a deep dive into the primary motor cortex, right? It's part of the cortex involved in, sort of in generating neural impulses, connecting with the peripheral nervous system, control of execution of movement and stuff. And a series of papers, so a lot of nice papers involving scientists here. Professor Aaron McCumel was an author, senior author in one of the papers. We'll talk a little bit about his paper a little bit later. And I know there's a lot of other things. This was kind of the cover. You could look. I would encourage you to go look at these papers to see and understand some of these types of data. And this was there was like a flagship publication that described the big picture, and then there were a series of companion papers that studied the molecular classification. Iran's paper, which studied a very nice paper, which studied the integration between transcriptomic, epigenomic data across many data sets. We'll talk about spatial transcriptomics. There was a paper on that, et cetera. Yeah. So anyway, go look at these papers. I'm not going to dwell on that too much more. These are a few slides here that came from that. So we'll go to 11 and take maybe 5 minutes at 11 o'clock. Yeah. People. Yeah. So, and there's a lot of, the techniques involved. We're going to talk as we go through these materials. We're going to be talking about some of these techniques in characterization. As I said, this is just the overview here. But now we can get really a deep kind of look at the structure, and so we have a really pretty good way of getting it. This is from Iran's paper here. This was kind of an approach to comparing. So in epigenomic data, there's different some of the earliest approaches that are really had to do with essentially looking for, so when you have the chromosomes that everybody, you all know this basic molecular biology, but to express the gene, the chromatin needs to open, it needs to be exposed so that the RNA polymerase can be active, it can actually initiate its transcription process and things like that. And it's not even so amazing. Maybe if you can even fathom that you could compact almost 9 feet of DNA into every cell, even if you're willing to imagine that, it's extremely difficult to imagine how the dynamics of how this folds and unfolds to expose these processes to actually allow functional transcription to occur. It's mind-boggling that this could actually happen. And so the epigenetics is largely concerned with the identification of these open areas of chromatin and their potential activity, right? The enhancers, the sort of basically kind of active kind of DNA active sites. And when we look at the patterns that we see, yeah. We have so now we have the ability to do single cell epigenetic profiling, like what's called ATAC-seq, right? Kind of transposon type of seq type things. But one of the troubles with it, it's a little bit noisier in a sense that it changes depending on state, probably more. It's a function of more things than simply the pure genetic programming of the cell. And so it provides really important information about what is likely to be active in the cell. But there's a lot more transcript gene expression data available typically than epigenomic data. Although now epigenomic data is being through either two things. One is discovering the open chromatin through ATAC-seq, and the other is a kind of discovering what states what base pairs are methylated, right? We know methylation is a change which occurs when a gene is about to be kind of expressed. And so we can do both of those groups that's here and groups at the Salk are, in fact, Joe Ecker, other people are Bing Ren, people are very involved in the development of these tools and technologies for methylation and applications of ATAC-seq. But it's being done in a big way, but it's still considerably less than the people that produce transcriptomic data. But both are very helpful. And interestingly, though, is that when you even independently kind of cluster these kind of data sets, you do see very similar groupings, which was a result of the Mukammel paper here. You see a lot of reproducibility of these types, which can be validated across these modalities. It's a very important finding. And why this is nice is because now we can start to potentially design like enhancers to actually, manipulate these cell types. We can build kind of genetic tools and they enable us to, for example, use them involved in translational or disease-related research, et cetera. So this is all very useful. For example, let's just take here in the Mukammel paper, right? This was the paper. It involved Zhizhen Yao in my group from the Allen Institute, she's one of our kind of transcriptomic data analysis experts. And the senior author was Iran here from UCSD. But this was the paper, it was part of the BICCN package in 2021. And they developed this procedure for really looking in a deep way of how to assign and how to cross compare these data sets, right? So, here, these were some of the steps that were taken. Just to give you an idea of some of the steps. We don't want to go into all algorithmic details. People are going to get, you get plenty of that. This is supposed to be more fun. So, but anyway, but we will be doing some of this. But, some of the steps here that they cluster the data individually, of course, right? And then the approach is that they find these cells which are identifiably kind of unequivocally distinct types, what they call anchor cells. These are cells that we know are actually unequivocally different types. In fact, we see them across the different data sets, right? And then we also, not all genes are kind of created equal when it comes to classification. We often look for genes that are differentially expressed, right? Very commonly. We're always looking for, it turns out that most of the anatomic structure of the brain can be deduced from the differential patterns of expression in the brain. If you have the most highly variable genes, you can see the anatomic patterns well. You can separate the cell types into groups well. You need this. You can't, if you don't have distinctions, you can't separate groups, right? And so that's where they use these. And then we, they compute for these, they compute nearest neighbors in each data set. And then they have a way of actually kind of doing a big kind of community clustering, right? Using, kind of community detection, kind of these Louvain clustering approaches. And then they put the pieces together by merging the clusters, reducing the kind of, the kind of uncertain ones, throwing out the noise, essentially. And then when you go, those are the steps. When you go back and do that, you see there's a really good alignment between these. These are, on the top here, what we're looking at are actually just like UMAPs of the resulting kind of clusters been colored by the different things. And these were a bunch of different data. The first group here the first three were by RNA taken out of the cell from whole cells. And then nuclei. Well, why nuclei? Why aren't the cells good enough? And part of the reason there is that the genes that are expressed and the RNA that's available in the nucleus is unfortunately even a smaller subset, considerably, maybe even only 10% of the RNA available in the whole cell. But as you go up into more, organisms that are closer to humans, it becomes increasingly hard to separate the cell from its extracellular matrix due to lipids and other issues like that. It's very hard to do this. And so people have had to develop techniques to bore in even more finely. In the case of humans, we typically work with nuclei, right? Because we can target those better than having to do all kinds of cell dissociation. Because when you do this RNA-seq, you've got to separate the cells from their kind of structural background. Kind of hard to do this sometimes, but these pictures show a very consistent kind of picture anyway. So these are kind of techniques here that are brought to bear on this kind of thing here. And then there's, like I said, the methylation techniques, ability to measure whether things are actually a kind of methylation is active. And another group, other approaches that are, so this is an approach here that's taken by a guy who's a really big expert in this, is a guy named Hongwei Dong from UCLA. He was actually in our group many years ago. He drew the original versions of the Allen Reference Atlas, our so-called common coordinate framework. I'm going to talk a bit more about that in a little bit. I'm not going to say later, because it's really annoying when people talk for 50 minutes and then they say, we'll get to that later. I mean, later. We've been sitting here 50 minutes. I don't want to go no later, okay? In a bit, how about that? So, yeah. So Hongwei's approach is, he's very interested in constructing wiring diagrams. And what wiring diagrams mean to him is that they look at anatomic regions. They use viral and other kind of techniques for tracing, for actually, you inject, essentially, many techniques are devised where you inject say, a certain virus into a cell, and it naturally propagates through the axon, right? Either in an anterograde or retrograde, away from or toward the soma. There's both techniques. And through this, you get an idea of what's connected to what, and you image it, right? And then you are able to essentially study these things. And it's not as quantitative as, this approach is not as quantitative as you might like. You know, they don't, unfortunately, it's a lot of looking at things and seeing what's connected to what and building matrices by hand and then drawing pictures like this, okay? So is it correct? Yeah, it's correct, I think. But it's also not something that it's easily amenable to analysis, data analysis, right? So we have newer techniques, things that where we actually attach kind of genetic labels, and they can propagate, the genetic labels can propagate, something called BAR-seq, right? Pioneered by Tony Zader from Cold Spring Harbor and others to do that kind of thing. So one big modality we haven't quite talked about, which is really, really important. So when we did, the first thing that we did at the Allen Institute, so when the Allen Institute was started, you know, Paul Allen he wanted to do something in, you know, he wanted to do a project in neuroscience, kind of motivated in the spirit of what the human genome project did, right? And he consulted a bunch of advisors. There was like a meeting on a boat, and they just talked about what such a thing is called a charrette. You ever hear that word before? Charrette? No, I didn't think so. Yeah. What's a charrette? Look it up. Charrette. C-H-A-R-E-T-T-E. A charrette it comes from the French, and what it means, I'll explain where it comes from in a minute, but it means when you collect a group of people, you have had charrettes, I'm pretty sure. You collect a group of people to brainstorm a certain idea and to talk about, you know, what you're going to do and what your plan is. It comes from the French, from the architectural students in Paris, that they had a part of their education was they had to do this kind of like project at the end, and they all would get together and talk about what their project was and their plan. And it was called, it comes from en charrette in French, meaning in the cart. So it was that they were like in the cart. They were kind of talking about their project. So a charrette is a word. If you look it up, it will be in the Webster Dictionary, and that's what it means. And so a charrette was had, and they talked about what we're going to do in neuroscience. And well, it was just, you know, people like David Anderson from Caltech and other people really thought that what we should do is we should build, you know, a large scale genetic map of the brain, right? And there was a technique that we had to do it. This technique, so-called in situ hybridization, right? In situ hybridization it's this kind of a technique. It's really it's almost orthogonal to like microarrays or cellular sequencing, because here you would pick one gene, you would design a probe for it, basically something that when you saw that you would know it, and you would attach kind of some fluorophore to it. Either it could be radioactive or it might just be colorimetric. And when this gene expressed, this would light up, and then you could image it, right? Or you could stain it, and you would stain. And so that's what we did in this Allen mouse brain atlas, right? We went through every gene, and we sectioned the whole mouse brain, not continuous sections, but 50 to 100 sections. And we took every gene in the genome, like 24,000, and we did this process, right? And we used robots to do it, machines. They weren't very sophisticated robots by today's standards, but they were robots. They called TCAN robots. They were like robotic laboratory things. And so it took about 3 years to do all that. Well, there's a technique now. So that was just one gene per section. And shortly after that, people started to really develop techniques for getting a couple of genes, using kind of fluorescent multiplexing and stuff. But far more recently, something that really has taken off is this techniques of like what's called MERFISH. This was pioneered by Xiaowei Zhuang from Harvard and some of her colleagues. But this is, and now there's very sophisticated versions of this. You can think of this as like the great granddaughter of in situ hybridization. In situ hybridization has been around since like 1980. That was like one of the first in situ hybridization experiments around 1980 maybe. But this MERFISH, now you can say even do 500, they're pushing it even beyond that. 500 genes, in which what you do is you design a combinatorial panel, right? And you don't, your panel is done such a way, it's using a lot of kind of combinatorics where you basically, you select sequences that you know are common to a set of genes, and then you do another disjoint set, et cetera. And then it's the multiplexing of all this combinatorics, which enables you to nail what gene is really active, okay? And so you can do this now. And when you do this, now you can do, say, for all these different genes, you can see in the spatial distribution of the pattern of the expression. It has some of the same issues of quantitation that the in situ hybridization does, is that it's still kind of a little bit, you're looking at, is something on or not on? And so if you really want a quantitative measure, you need to go back to the RNA-seq, right? But it does provide you with the spatial context, which is essential, because now we know when we see certain sort of cell types, you know, if you really want to solve the problem, you want to know where they are, right? For these kinds of, you know, cell types that we call layer 2, 3, you know, IT cells, that they really they live in these layers, laminar areas of the cortex, right? So these MERFISH and its techniques are an important part of the puzzle. And we spent a lot of effort to map all this data into what we call, a term was evolved to be called, I don't know exactly when this term occurred, but in my career, this term has come to become accepted, but it was called common coordinate frameworks. And I think I might have been in the room the first time it was pronounced. I can't remember, but I don't remember where or when it was pronounced, but it basically means, so what you want is, if you want to understand the structure of data, it helps to have a frame of reference, right? And it turns out for the mouse, this is not that difficult compared to, say, human. And basically it's one special mouse that you deem is going to be the important one. And how this mouse was selected originally, well, the way that when we did the original reference atlas, the way it was selected was rather because some people sat in a room in a cryostat and cut hundreds of brains until they got one where the sections didn't fall apart, okay? Probably took hundreds of brains to get a good series, because it had to be done on kind of, it had to be done in fresh frozen tissue. It would be easier if you perfused the tissue, right? If you essentially solidified it, but then you would distort the structure and the relative relationship of the areas of the brain. And then when you tried to map data to that, it wouldn't be accurate. And so you want your reference atlas to be constructed in the same way that you're going to be doing your data collection. And so we did this, and then Hong Wei sat in a room for 2 years, and he drew those pictures, he drew those things. And now I don't know how these anatomists do that, because, you know, they look at this, how do they do it? Well, what they do is that they take the section and they use a stain called a Nissl stain. You've probably heard of that. It was due to a sort of a biochemist named Franz Nissl around 1850, something like that. It was a way, and the Nissl stain largely stains all neurons by staining certain aspects of the endoplasmic reticulum in the cell. It targets that, something called, what they're called Nissl bodies. It can stain them. And you don't get a really clear shape of the neuron, but because it can be erratic because of the erraticness of the staining of these Nissl bodies, but you get a pretty clear idea of what's a cell and what's not. And so the neuroanatomist, a neuroanatomist is someone who can look at that and see patterns. Now, you know, it baffles me. I mean, sure, you can see the cortex. Any person can do that. But, you know, I can remember when we first started doing this, this was before deep learning even. You know, this was like, I remember what year it was, but before any kind of deep learning really took off. But I can remember some friends from Microsoft Research came over and they said, you know, well, you know, never mind this drawing, just send us the data. We'll learn the regions, you know. I said, you're not going to learn the regions. It's a voodoo. I don't know how they do this. They look at these patterns and they see these things that You're not going to do this. And so, as it often turns out in academic collaborations, no one ever calls you up and says, you know, Sally, this didn't work. We're having trouble. We didn't get anywhere. No one ever calls you back. They just don't call you back, right? So, and so that's what happened. We never heard back from them again, you know. But anyway, so they draw these things, and it's not just based on the Nissl stain, but a neuroanatomist has a lot of extra insight. They know other cytological stains they can do. They know the historical context. They know the connectivity, and they use gene expression data to draw to see regional boundaries. And so that's how it's originally done. More recently, in our present version of this, we use kind of more like a tomographic kind of high throughput 2-photon kind of things where it's done with a technique we call it like block face imaging, where you essentially take a picture, slice, take a picture, slice. And if you do this really carefully, there's machines that can do this really carefully now. You can reconstruct it largely tomographically, just like an MR machine would do to get a true kind of 3D thing. And do this, you really get an unprecedented resolution. Here we're looking at an average of about 1,600 of these images, okay? And you see this little patterning right here. Well, this are actually the barrels of the barrel cortex in the mouse, right? The barrels, these are areas that are really connected to the somatosensory kind of functioning of the mouse. Whiskers, control of the whiskers and other areas. Things that show up in averaging the averaging has a kind of a purifying effect, right? Sometimes in data that it actually can highlight what's really there. Now in the human, it turns out this is much more difficult to build common coordinate frameworks because of the variability. One, we don't have the ability it's bigger. It's 1,000 times bigger than the mouse. Two, we don't have access to human brains the way we do mouse brains. Three, the variability is phenomenally. We don't have inbred, like we have these inbred mice. We don't have, fortunately, inbred humans that we do that on. So developing common coordinate frameworks for the mouse is at work that's underway. There's been a lot of progress. Many people have done this over the years, and we continue to evolve that, but it's a trickier problem. So I think let's see here. This was just lecture 1. I think maybe we'll take a break let's take a break for a couple of minutes, and then we'll continue. I think we're gonna. Yeah.
Whether there's a reference of like this diagram regarding other areas like this.
People have people done this for approaching the hippocampus?
Or some picture like, yeah.
Yeah, I don't have a picture of it, but certainly, actually my question was the previous slide. Okay, all right, let's go back. This one. Right, thanks. Okay. Mingji, yeah. And so what is the question? Oh, do we have such a circuit diagram in the hippocampus? Yes, people have done that. I don't have access to that, and I can't at the top of my head provide you with a reference, but the hippocampal kind of structure, architectural structure for between CA areas and Ammon's horn, et cetera, the dentate gyrus has been kind of elucidated doing this. It's been active of a lot of study. You undoubtedly can find such papers. I could probably give you a reference on that, but I can't off the top of my head. But it certainly has been looked at. Yeah. Okay. All right. Thank you. Okay. Let's stop here. I'm going to start. We'll start on this slide in a minute. Let's just take like 5 minutes and stuff like that.
Some different kind of clustering. How do you even decide what is all of this unsupervised, right? So how do you know what would be a good outcome?
Yeah. How do you know? Well, yeah. What's the validation?
What's the validation?
Well, so the validation, we don't have in most cases a functional validation like the ultimate validation is this does something. And first of all, it's science. So science is always subject to revision, right? So even if we knew a type and we said this type appears to be active in this, we might find later that it had enhanced function, right? Or we might realize that it was doing something else. So it's a bit of an ambiguous, it's not well-defined question in some sense. But the way, from a data science perspective, the way we do the validation is, we're going to talk more about that. We'll talk more about that. But the basic idea is that either you do resampling techniques, right? Reduce the data set, do the same operations, do hold out, etc. You know, cross validation, yeah, or you do comparative other modalities, comparing epigenetic clustering to the transcriptomic clustering. You can't really validate very well transcriptomic data using other modalities like morphology or electrophysiology too well, because you certainly can't discover a new class because the variability, there's not super well correspondence. It is, transcriptomic type will have a variety of morphological. Once you know what you're looking at, you can make the association, but you have to use kind of more standard data analysis techniques to just do that. It's reproducibility in the data. Yeah. So, is it a helpful?
There's something like accepted ground truth for which type of data you would prioritize as being like if you get a bunch of different clusters from
It's a good question. That's actually an excellent question. The question, in fact, let me tell you how important that question is. In fact, it's at the heart of a lot of discussion and debate. The classical neuroscience were more from, and largely due to the unavailability of molecular techniques until more recent times, we're much more concerned with neuronal structure, shape, form, and then reporting. And so they would have tended to prioritize. When the molecular kind of revolution sort of took place, the people who are champions of that are rather dismissive of anything but molecular methods. They tend to feel that I'm kind of a little bit unbiased with that, but they're a little bit unbiased. They tend to feel that the molecular approach is really the most it's the most quantitative, it's the most reproducible, and it's the most unbiased. And so they heavily prioritize that. And they have largely, due to the amount of technology behind that and the truth in some of those statements, they have largely won over the classical people. But the classical people is really happening. The classical people still always agree, they say, but we need to consider the morphology and this, right? And the transcriptome people know that, and they admit that. Okay, they agree that the cell's true characterization is not. It can't be done without these other types of things. But I would say the field has become overwhelmingly driven by the transcriptome, largely due to the transcriptome.
Do you think the limitations of how transcriptomics are data is collected now is what is kind of creating this barrier between identifying cell types based on morphology versus transcriptomics? Like if we had better transcriptomic methods, could we capture morphology in that data set?
I don't know. That's a good question too. Let's take that one too. So the question, your question really is what determines the shape? What determines the morphology? And it's sure, a thick tufted pyramidal cell is largely this genetic program which determines the fact that we can run that. Question one, we don't know what that is. That's a big developmental issue. If we can discover it, we might not have to look at shape, we would know it, but we don't know it. And so for now, we want to collect both to kind of get the thing.
Yeah.
Another thing too is that another problem with that is that it may be, as you know, that genes will be active at certain stages of development, and then they will turn off. And so the result of the genetic program is there, but it may not be accessible to you. use and in the adult. Okay? You may not be able to, sure, it's something once upon a time determined this was going to be a TikTok or grandma, but you don't have access to that information now, right? And unless you do study the full development of the series. Right?
And I think a lot of external factors are around things.
Right.
So that sounds like to me there's like some involved.
To get the real story, you want to do a full development series, right? You want to, it's really about that. And there's efforts, big efforts underway. There's a big effort underway now to do a fairly deep time series in the mouse. A fellow from University of Hawrylycz, Arnold Kriegstein, I don't know his name, but he's a big guy in human developmental thinking, model techniques. You look up some of these people. Yeah, so no, it's development really matters, okay, right? And yet, I think it's a matter of time, resources, money. So people, they start in the adult. The adults are doing the work, so they probably prioritize the adult. They're more worried about themselves rather, so but people, there's a lot of work happening for sure in this, in enormous amount. Children from many children's hospitals. It's a lot of oh. Yeah, I mean, that is. One line, maybe. If it was big, you're gonna be able to see where it goes. Exactly. Okay, we're talking. Yeah. Okay, we're back. Okay, so let's just finish up this little unit here. There's only just a couple more slides. A couple more points I want to mention in this unit here. So one of the things that makes the mouse easier from a common coordinate. So what's the purpose of these common coordinate premiums? Common coordinate is to have a data structure that embodies and describes graphically in images the anatomy of the brain, to have it be a sufficiently high resolution that we can align using image processing methods, images to that, either sectional images. So the big problems in image processing, what are the two big problems in image processing that dominate every other thing? Segmentation, right? Finding something, determining what's there, segmenting it. In our case, it's segmenting cells, segmenting neurons. We'll talk about some of those strategies a little bit later. And registration, right? Aligning things. Those are overwhelmingly, the biggest problems probably in image processing, especially in neuroscience, But in the case of common coordinate framework, it's more about registration, and an enormous amount of technology has been developed to do this. It's, you're aligning, there's a lot of techniques for aligning like MRs to MRs, right? They do Lucas's shoulder back there. They probably they could align it to a general MR shoulder and say, how bad is his shoulder? They may not have done that. I'm not sure what his health plan is like. It may not be good enough for that kind of it. But that's the idea, And but in the case of the mouse, as I said, because of the homogeneity of these mice, by averaging over a lot of them in images, you get a very clear picture. Like here, here we're looking at, this is the hippocampus right here. Again, this is all cortex. This is the thalamus in here. But the mouse brain, it doesn't hardly have any folds in the cortex. It's what they call lissencephalic, meaning that it's largely flat, right? And the human brain is, of course, has all the sulci and gyri, which we believe, it would enable the compacting of a lot more cortex into a confined space. Some people believe that there's theories about how communication has been improved through the proximity of things, et cetera, like that. But in the case of the mouse, you have this largely flat lissencephalic cortex, and you can segment out here. Here we have segmented out the different layers, right? We know that all mammalian cortices mostly have 6 layers. Okay, it's a known thing. Mice do, human do, et cetera, right? Yeah. But the nice thing is that you can do, because of this essential flatness, you can build a flat map of the cortex. And we do so by, using kind of Laplacian methods, and we construct streamlines that go perpendicular to these kind of layers, and we can flatten it out in silico, okay? And actually, people who study the human cortex, like David Van Essen and these people from Washington University, people who really study the cortex and function believe strongly that you really should be working in the cortex with flat map. And they use either, how do they do that? They either use, for software, they either inflate the cortex, right? Or they use techniques to kind of flatten out they can inflate it to a sphere, or they can actually use techniques to kind of unfold it. And why do they do that? Well, because what is proximity in a 3-dimensional sense of like where the motor cortex is here compared, let's say, to the visual cortex may not really be close in some sense. You better see it when things are flattened out. So in the mouse, in this common coordinate frame, we do that. We have flat maps. We study data in those ways. We have tools and techniques. There's a set of Jupyter notebooks online. If you go you look online, you can see some Jupyter notebooks that enable you to use the common coordinate framework and to map data into flattened context there. Another thing which is nice about going back, this is going back to the transcriptomics here. Another thing that's nice about that is this ability so if you think about genome mapping, right? So one of the major things, the way that we've learned a lot about the evolution and a kind of development or historical development of organisms and things like this is through homological mapping of sequences, right? Sequence alignment. In the earliest problems of like computational biology were, and when I was back at Los Alamos, I was in Los Alamos for 5 years, 1992 to 1997. There, chromosome 16 was a part of the Human Genome Project. Chromosome 16 was sequenced at Los Alamos. So I was there when we were doing that, worked on that a little bit. All the problems about computational biology back then were concerned with sequence alignment. That's all it was, right? Because the way they did this is that they used these so-called shotgun approaches. We couldn't get big, you can't get infinitely long sequences. You can get short groups. And what you do is you get coverage, and then you use techniques of string alignment, right? Different kind of dynamic, you probably study dynamic programming, right? And you use techniques of dynamic programming and string matching to typically align these sequences and to find out how to build the thing. Well, so now if you want to classify, so when you classify like cell types transcriptomically, what are you doing? We'll talk more about this in a little bit, but let's just do the superficial version first. Is that what you're doing, right? Is that you're sequencing RNAs. First of all, there's a small amount of RNA in the cell or cell nucleus. So you amplify it, right? You use PCR or something like that to basically multiply it up and essentially blow it up to something. That has its own problems. It introduces problems of doublets, other things that come about, and idiosyncrasies in it. But you get enough that you can measure. And then what these machines do is that they take these sequences, they take these oligonucleotides, and they basically use kind of laser type techniques to determine what is the base pair, based on its molecular weight or other things, and they string them together, and that's you get this. Now that you've got that, you can read out its sequence of nucleotides, CA, C, T, T, C. So what do you do with that? Well, what you do is that then you take your map that you had from the Human Genome Project or any number of organisms now have been sequenced in the database. You go to National Library of Medicine or GenBank or something, and you use techniques like BLAST to do sequence alignment, which basically find the place where that string aligns, right? And that has taught us an unimaginable amount about biology. The most cited paper in all of biology is the BLAST paper by Achul and Gene Myers and others. That paper, it's cited like 120,000 times. And it's because everybody used BLAST. BLAST was like of fundamental importance. It enabled people to align strings. It's taught us about variation between organisms. It's taught us about, we can identify polymorphisms and areas of risk genes. And we can sort of do things like map between species in a cell type way. So how do we do this? What's the analog? So when you align sequences, like you might have so when we, for example, we want to determine whether something is a gene. It's largely determined through annotation. We don't conduct experiments to show that it does something. Usually we don't know, we don't have any idea, but we align the sequence to known other things, and we deduce, well, this must be doing something. And then we have further evidence to show that it produces, a transcriptome or something. But so this kind of homology kind of alignment teaches you a lot. And so in a cell type context, we can do something similar. What we do is that we first build individual clusterings and get individual taxonomies of like a mouse and a human. And then we get the marker genes or genes that are differentially expressed, which determine, they say, oh, cluster 7 is really different than cluster 25 because I can tell you these genes that are high here and low here, right? And so we have evidence of a distinction in cluster. And so then we have such a taxonomy for the mouse, such a taxonomy for the human, and we align them by showing that they're not necessarily the same genes, but there exist genes which differentiate clusters to clusters. And this produces a kind of homology mapping, which you can see here between like mouse and human versus marmoset and human. And generally, what you're able to do is that you can't take this down to the thousands of layer kind of clusters because it becomes noisier and noisier as you go down. But we see down to a pretty good level of this very good alignment between these species, right? And this is important because it allows us to do some things like we can start to study, well, what genes determine this cluster in the human and what do they do? And this must be an interesting cell type because we find it reproduced across species. So in the absence of functional information, this kind of homology mapping gives us, kind of a foothold, right? So we do a lot of that. And we've done this. We published papers on this and stuff. We did, here's a paper that we published with a guy came and visited me from Montreal. And we did this paper. This was a study of disease risk genes between kind of mouse and human. And so what we did was we aligned these taxonomies between mouse and human. So we had clustered the human, clustered the mouse. We did this alignment procedure. Here we saw how the cell types, this was like a kind of confusion matrix about how well things map between the two. If it's along the diagonal, things are going pretty well, right? And then what we did was, so here you have about 40 or so major brain diseases. And so we went and we get the diseases that are known to be associated with these diseases using any number of disease-related gene databases. And we can show, so the mouse is in orange, and the human is in blue. And you can see these different diseases here. And on one thing, let me see what we got here. Yeah, so on the left, it's basically what we're showing is that if we cluster what we're doing is we're clustering the concentration of gene expression in each of these are kind of a major set of cell types here. And so we're saying for these disease risk genes, so let's make clear, this is not done by people with these conditions. We don't have access to that. They're simply done by genes known to be risk genes for these conditions, okay? A lot of available information about that. But when you cluster these, what you see is that if you cluster them using the cell types that they're involved, you see that the diseases have an overwhelming similarity between the risk genes between species. But when you look at the actual expression levels, the expression values, we see that they're much more species specific. Namely, that the same cell types are at risk between the species, but there are different genetic programs that may be active to be doing that. It's a basic analysis. It's not curing any diseases. I'm not trying to claim that, but it gives you some insight into some of its activity. So, one of the goals in all this stuff is to build these reference taxonomies and spatial references, right? What would be we're not there yet, but what we would like to do would be have something like this. It'd be a web page on the web. You go to the web, and this is what you see. You see this really cool kind of like circular hierarchical diagram like this, and you'd be able to kind of like touch on any cell type, and you'd be able to see, let's say, it's the cell types that were active between the species. You'd be able to kind of get an anatomical function. You'd be able to see for that cell type using the MERFISH and the spatial stuff, where are these genes expressed in the brain, right? And so that's what we're trying to build. We're trying to build such a thing. We have preliminary versions of it. We have something that we did called the Cell Type Knowledge Explorer. If you look it up, CTKE, Cell Type Knowledge Explorer. We combined all the data. I think I have a slide on it, but unfortunately, you know how it is when you build a talk. The slides are never in the order that you're talking. You try, but we'll get it. It's in there. There's something called Cell Type Knowledge Explorer. And it basically combines all the data from that primary motor cortex study from the BICCN. Remember the one I told you about? There was a package. Combines all the data, all the different measurements from people there. And it basically puts them in a taxonomy, and you can browse it and you can get the data. Yeah, so that's pretty cool. Recently, we released, so eventually, like I said, you have to declare victory at some point. There's finite many people they want to declare victory because you got to move on to something else, right? And so largely victory has been declared in the mouse brain from a transcriptomic perspective. The BICCN has finished. Does that mean that we're done with the data analysis? No, we're not done with data analysis. Efforts are on the way, right? There's a lot of opportunities there. If you're interested in this kind of thing, interested in transcriptomic data analysis, there are innumerable projects that you could get involved in. We just released this kind of a big atlas here. So we finished this transcriptomic profiling of the whole mouse brain with 4 million cells using RNA-seq. with 9 million cells using MERFISH and transcriptomics, it produces this unimaginable number of 5,322 hierarchical clusters. Don't ask me what they do. I have no idea. But they're arranged, again, hierarchically into things that we're more confident about to less confident about, right? And with this kind of dual approach using the RNA-seq and the MERFISH, you can, for example, touch on, this is like a big UMAP organized in according to colored by the subclasses. Okay, some level of resolution is colored by that. All the ones that are of similar color are cells of a certain type, okay? This might be parvalbumin expressing here. You know, Lucas is gonna probably keel over if he hears parvalbumin one more time, but that's you see, that's the kind of thing there. But anyway, when you touch on that, you can go over to the spatial side and it will show you where those cells are actually in the brain. Okay, that's the purpose of this atlas. It's on. You can call it ABC. We call it ABC Atlas. You can go play with it there. We've also applied this in a translational work. We have a project called the Seattle Alzheimer's Disease Project, CAD. It has its own website, CAD, I think something like that. Seattle-AD. And we basically built. This is, again, this alignment activity I'm talking about. Here, we built taxonomies using cells in the middle temporal gyrus of human. Middle temporal gyrus is a highly accessible area in the human cortex, largely because it's an area which is often resected during epileptic surgeries, okay? So you can get that pretty readily. It doesn't destroy brain function to take out pieces of it, like some areas might. And then we collected data from a number, about 90 or so Alzheimer's patients at various different degrees, stages, and you know, who were deceased, and basically what we did was that we studied the cell types in one. We tried to determine if they could be found in the Alzheimer's thing, and we augmented our taxonomy to reflect those which were disease state related. Okay. And so this is the kind of thing you can do in all this. And we built a viewer where you can see this. Okay, you can go there and see this thing. And this data is all available. You can get all this data, do further analysis online. You know, of course, the epigenomic data is about what epigenomic is typically, at least from the attack-seq is, again, about, as I said, that's about exposed chromatin. It's finding areas where the chromatin is open and transcriptional machinery is potentially available. The earliest techniques of that were things that were kind of, what they did was that they were kind of basic assays, but what they did was that they essentially, there was a certain kind of enzyme called DNase 1. And this is a special enzyme that what it just wants to do, it just kind of cuts, right? When it sees a piece of open chromatin, it will tend to digest that and it will tend to separate it. And so you apply this en masse to a large set of dissociated cells. This is before single cell things. And it would go through and produce all these cuts. And the cuts were evidence for areas of open chromatin because they were placed, to cut it, it had to be exposed. And so then what you do is you would sequence those, and then you would map them, and just like when you look in a genome browser, what you're typically looking at is you're looking at some form of tag pileup. You're looking at where something is active, where tags have been where they've been mapped. And so this is how we map chromatin accessibility, typically using these tags. The computational problems involved here are where one is, you're typically interested in determining peaks in data, right? I wrote one of the original ones for this a long time ago, something called hotspot. It was one of the first epigenetic kind of technique. We did it a long time ago, but then it's very primitive compared to what people do today. This was like a long time ago, but it basically went through, and what it would do is it simply, you can imagine here's how I abstracted the problem. Imagine you have this landscape just like this, right? And so there's going to be, and you can just think of it as that the genome is here and it's all aligned. And every time you get a tag, it's going to start at some base pair. And so I'm going to just record that, right? And I get this kind of like Bernoulli process where things kind of just tags pile up, right? And now I have a certain sampling density, and I can start to look for regions subject to certain windows where there's an statistical over-enrichment of tags that are piling up, right? And I can look for regions of high variance. I can do this. And that's what Hotspot did, is it basically scanned through looking for these regions of enhanced pileup, and it ranked them based on statistical significance, based on the number of tags which were thrown and the size of the region, okay? So that's some version of that is what they do today. It's more sophisticated, but that's the spirit of it, you know. I talked about mapping cells to references. We have a tool now that you can go online. We call it Map My Cells. And if you have a set of data, you can, your data would be given in the form of like a FASTA file or something like that. It would be basically a list of sequences, and these sequences would just be genes that you knew or believed to be expressed, and you import them into our thing. And we have different mechanisms for what we do is we try to determine in that taxonomy which are the cell types that are representative of the data you put in. And we do a couple different ways. One is a simple way of like hierarchically marching down the tree of saying, oh, look, it's closer to this one, correlate by correlation, et cetera, bingo, 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 like that. But then we also have a more of a variational autoencoder way. I'll talk about that later as we get, I say we're going to get more quantitative. We will start getting more quantitative, but that's, there's an implementation of this variational approach to map it. And it seems to do a little better than simple correlation. Yeah. So, but, the cell type knowledge explorer, here it is. Right. So you can go check it out. Yeah. This summarizes all the information from the primary motor cortex in 3 species. And it has all the different things you can, it's got the patterns, it's got the transcriptomic cell types, it's got the morphology. It's what we know and what we discovered in the data from that. And like I said in the beginning, so I wrote this paper maybe last year or something like that, but the paper was I was motivated by a paper that was published. This is a paper in 2011 when ENCODE had first done. I was involved in ENCODE in the beginning, but then I left when I did other things. But ENCODE is kind of more or less done, but it was for a long time produced a lot of data about profiling the regulatory DNA active functional elements. A lot of people contributed to this. It was really a big effort. It stands for Encyclopedia of DNA Elements, right? And so they wrote this paper, the consortium wrote this paper, it was published back in 2011, and it was a user's guide to it. And so I was motivated by that in producing a similar description of our BICCN data. And so got all these people together, and everybody contributed some kind of, either vignettes or something. There was an enormous number of people in this ecosystem of the BICCN. And so this summarizes the state of all that kind of work in that paper. It's a PLOS biology article. So, okay, so that finishes unit 1. That's our kind of overview, okay? We're gonna go into some more details about different things. And I think that we're gonna have to, I said, as of course it's going slower than you want. I have actually, that was 7, we have 6 more to go, but we're not gonna finish 6 more. So we're gonna have to pick and choose. But you can always, you'll have all the slides and you can do it. So let's go here. And first of all, do we have any other questions or discussion about this first segment here? Anything else that you feel was unclear or something you want to know a little bit more about? Or
for each cell types. For example, if I want to figure out a specific gene that is expressed for specific cell type for labeling purpose, and we check those information in Atlas.
yes, you can tell. I mean, we can tell. So it's a different question because. Oh yeah. Over here. So we can tell. So the question, we have to go, let's back up a tiny bit on that one. What was the question? The question was, if I understand the question, the question was, if I have a gene's pattern, can I tell what, whoever asked that question, I want to see that you say yes to my question here. Okay, I'm asking you a question. The question is, was your question that if I have a gene and I know its expression pattern, can I tell what cell type it's most associated with? Is that your question?
It's in the chat window.
Yes.
Yes.
Oh, well, let's say that's your question. Okay. That's your question. You're going to get that answer anyway. Anyway, so the question is, yeah. And so this is the thing that where complicated is this, is that what determines what's transcriptome? Here, we're only talking about transcriptomically. What determines whether something is we got a yes, good. What determines whether is something is a cell type transcriptomically? It's unfortunately not, it would be nice if it was like one gene per cell type or two genes that uniquely nailed this. And it's usually not the case, right? It's usually some combinatorial pattern of expression which occurs, which differentiates one from the other. And there's variability in that, as Lucas knows. There's mixing and matching. If you were to resample the data, you would see some crosstalk, okay? If things are really separated, then generally you'll get separation more often than not, but sometimes it becomes more ambiguous. So given a gene's expression pattern, we can do a couple things. One, we can determine whether that gene formed one of the markers that was involved in the panels that was separating cell types. We can determine that. We could determine that gene's contributory effect to any given cell type, right? In other words, how much of its expression is in each of these types. You can do that, yeah. Okay. Thank you. Okay, Minji, good. All right, so let's go back to the beginning here. So the next section here is a bit on cell morphology and connectomics. And I don't think that we're gonna, I think I just want to show you a couple things in that. I don't think I really want to, that we want to do everything there in great detail, because then we're going to miss out on some of the cooler computational stuff. But there's, I mean, some of the other stuff that may be thematically more interesting, but I want to let me just, there's a few points I want to make here. Okay, let's keep going. Let's keep going. Okay, I'm going to go a little, I'm going to start going a little faster. Okay. Not talk faster maybe, but I'm going to start going, whoops. I'm going to go a little faster, meaning I'm just going to spend less time. You're already kind of semi-experts now on cell types, the modalities. So now by now, I think we understand the major modalities. We understand some of the issues with the modalities, right? We understand how they contribute to cell type definition and what some of the shortcomings are. Okay, I think, can we say that we're there? Okay, we're there. Okay, let's talk a little bit more about some of the details of morphology. I'm going to kind of blast through this a little bit pretty quickly because it's not. So basically, again, as I said before, the main issue in the morphology is that, so these are the kind of patterns you tend to see. The soma and up here and the dendrites will tend to be locally in one layer type of thing. And oftentimes pyramidal cells, a major type of excitatory cell, will project kind of upward. They'll project their so-called apical dendrite or their axon up to a more superficial layer or higher layer kind of in the cortex. This is very common. And you can typically often characterize patterns in cell types by where the soma and its dendrites kind of live and where it's projecting to like that, okay? This, again, it doesn't tell us though, we talked about this, that this is kind of this mesoscale approach where we get the local morphology or we don't get this, yeah. Let me back up a tiny bit. In connectomics, we want to think of 3 layers of resolution: a micro level, a meso level, and a macro level. Let me define the modalities involved in each of those, okay? At the micro level, it would literally be electron microscopy. I'm going to show some of that. Reconstructing detailed arbors of the neuron, okay? At the macro level, what's macro connectivity? That's fMRI. This region of the brain gets more blood flow when you are reading a book, okay? That's pretty macro, right? It doesn't tell you anything about what neurons are involved. It just says a whole millions of them are doing something when this activity occurs. And there's a meso level, and the meso level is about, you do get projection. So we built a connectivity atlas some years ago, around 2009 or something like that, in the mouse. It was all done by using transgenic mice, injecting these tracers, following them, imaging them, mapping them using registration to the common coordinate framework, and then looking at what region was connected to what region. Why do I call that mesoscale? Because we are gone beyond, we do know that certain neurons are connected to certain neurons, and we do know that it's going from one region to another. But so we've beat out the macro people that way, but we don't know which neurons are connected to which neurons. We don't have a circuit, okay? All we know is a bunch of neurons are communicating with a bunch of neurons. And that's what I mean by mesoscale. So we built a big atlas on that. This is so-called the mouse connectivity atlas. You can see when you do an injection, what was continued, what are the clusters. All this data is kind of available online. And you can come up with organizations of, we did studies, you can come up with organizations of circuitry. We know that there's a lot of feedback between the cortex and the thalamus, right? Thalamus is, as we know, a kind of relay station. Information comes in from the eyes. It goes through the thalamus before it goes to the cortex, before it goes to the visual cortex. It also has a lot of activity mediating cortical function and helping different areas of the cortex talk to each other, okay? So we studied some of that architecture. This was all done by connectivity, it's not gene expression, right? It's done by what's projecting to what, okay? So what are some of the problems in construction? Let's talk about some of the problems in that, like computational problems. So prior to really the availability of really deep learning, the way one approached morphological reconstruction was we approached it by classical image processing segmentation. What are known in the image processing community as morphological operations, right? Operations of, what is that? It's like you see, if you've studied image processing, you know exactly what I'm talking about. But it's basically like separate a ring from its outside. Filter this thing by dissecting it into a bunch of squares. Such operations like that, performing symmetric difference of two patterns on an image. These are called they call these morphological operations. These were all the building blocks for how we did image segmentation prior to the emergence of deep learning. And it didn't do a really good job, okay? It does an okay job sometimes. And you basically, because the problem is that here was the problem, is that typically when you got an image, it would be, this on the left over here is an example of an image of a biocytin filled section. And you see that, and a human can look at this and say, oh, I see the dendritic patterns and things here. But the trouble is when you blow it up, sort of at resolution, maybe this 100 micron resolution, what you see there's all these gaps in it, right? These gaps, and things will break. And they don't really break, they're just breaking in the image, right? And so when I use the classical morphological image processing techniques, how do I do it? Well, what the algorithm would typically do something like this. It would segment a continuous body based on some watershed method or thresholding, right? Watershed methods are when you change levels of a function, and you're essentially looking for active versus non-active regions. And but then you were faced with this problem of having to connect these globs, right? And to connect the globs, well, globs could go like at weird angles. The gap might be bigger. It was a mess. And so it only produced not so great region. And so people would do a lot of manual tracing, would look at these things and through the microscope and trace them all out, Well, convolutional neural networks have really kind of come to the rescue. They're immensely powerful. The number one successful application one of the major in neural network methods has been in image processing. It's a revolution. It determines the thing. It really enables us to see patterns that we inherently see, but we can't really get our hands on them except by using that. And we do it by doing a bunch of manual traces. The manual traces do things like the manual trace becomes the training set versus this. So the machine learns to understand what a gap was and when the gap was filled. And so when it's put through the neural network, like using UNet or something like that, it can actually cross these gaps and produce very nice kind of segmented kind of things, right? This is done examples are done in light microscopy data, right? This has its own set of challenges. One is that luminance issues of whether, something was actually there captured. And so we do a lot of analysis of that. I don't want to go into too many details. You can see there's a lot of, clustering, whatnot. You examine this data, you study and stuff like that. But, through these kind of things now, we use these methods and we get much better shapes. We get much better patterns of morphological structure. There's a paper by Olga Gliko in my group, and she applied these methods to doing, to doing these kinds of clusterings and analysis. So it's very powerful, these kind of approaches. This is a pipeline from, we were talking about Han Chuanpeng, his group. Han Chuanpeng's group now, they do a lot of machine learning techniques, but he was one of the real main leaders of also the pre deep learning approaches to segmentation of neural things. A lot of his packages, original packages like VA3D, were based on using more classical morphological operations. But now, of course, they're doing more stuff like that. So this shows an example of the VA3D pipeline, how it works, and basically able to segments data, maps them to CCF and stuff like that. I want to skip all this. This is just there's more stuff about. So there's a technique now. So to get the long range axonal projections, we now use a technique called FMOST, right? And this is a way of basically what it does. It clears the brain, and enables you to focus in on essentially a very, very localized set of stained kind of neurons. Okay, and so now you can get their long range projections and you use a cleared brain, and then you image the whole thing, like using these tomographic methods of that, and you can reconstruct them. And now you get to see the long range axonal projections. These are very important for determining the cell types, and they can be mapped. Here's the example of using the FMOST technique. These were cells that were in the claustrum. It's a strange area of the brain right below the cortex. We don't know really what it does. Some kind of mythological people think it's the source of consciousness, but actually, it was Francis Crick who proposed maybe it was the source of consciousness, and Christoph Koch kind of followed him, but we don't really think that's the case anymore. It's kind of centralized area, which is apparently connected to a lot of areas, and so it attracted attention. But so here, this study did a detailed study of neurons originating in the claustrum and where they projected to. But it was using this FMOST technique together with the cleared brains, a technique developed by Carl Dieseroth in his lab at Stanford, and then I'd be able to do this long-range mapping. It's really cool. I'm going to skip it. I want to talk a little bit briefly about electron microscopy. So there was a project that started maybe 10 years ago now almost, but it was called Microns, Machine Intelligence from Cortical Networks. And this is a project, it came from IARPA, which was, it's kind of like the Intelligence Advanced Research projects. It's a relative of DARPA. You've probably heard of DARPA, which is defense-related projects. IARPA was kind of more of an internet-based kind of version of DARPA, but certainly with a military interest, right? And they formed this project, and the idea was that, what was the idea? The idea was that if we really did a deep dive and understood the real connectivity, this is micro connectivity now, right? We're talking exactly the shapes and structure of neurons and exactly what they're connected to at a synaptic level. Now we're going to be looking at synapses, right? And synaptic gaps. And using electron microscopy, which is immensely time consuming, expensive, but very good for very local kind of data. We and others were involved in a project that basically essentially profiled like a complete cubic millimeter of the mouse cortex. So massive data set, petabytes, right? Sort of a workflow. There was a workflow, started with kind of functional imaging. We actually did a mouse in which it was a genetic mouse in which certain cell types were active because we wanted to know what was happening in this little area. Then it went through sort of things, some chop into blocks, some like sectioning, et cetera. And then it gets put through these kind of, electron microscopes, which now have a resolving power, far greater than light microscopes. But now you're confronted with this immense computational challenge, and that's of reconstructing, right? And again, what really comes to the rescue here could not be done using classical morphological techniques. It would have to be done using deep learning, right? And Sebastian Sung's group from Princeton were really the leads on this. And so they use, let's see if we can, this is an example here. Let's just take a look. This is an EM, we're looking here at an EM block. Now, so these are thin sections. They are nanometer sections now, like 1, 2, 3 nanometers, right? And if you want to use this data and to make it so that you actually can follow a process, you cannot lose more than 1 section contiguously. If you lose 2 sections, you may lose the thread, okay? This is an astonishing thing. So here we're boring into this section. You can start to see the emergence of cells. The cells are starting to come about, and as we go in deeper, we're going to start to see the patterns of the cells. But you see, this is looking just at cross sections. You have no idea from looking at this what is the actual, and what seems to be moving, of course, is just the path. It's the path of some neuron's body traveling across through this structure. And we're going to see in a little bit how these are pulled out. Pretty incredible, it's pretty incredible. See how far along are we in the movie? Oh, we got along with it. Let's advance this a little bit. This is getting boring. Yeah, so the trick now is that given this, let's say, given this was what you see on a section by section basis, how do you reconstruct individual neurons and separate them from that, okay? And using this kind of pipeline, it was a combination of the main participants in this were Princeton, Sebastian Sung's group, Andreas Tolius from Baylor, and our group from the Allen Institute. But the reconstructions using the software that was kind of pioneered in Sebastian's group, it produced like 84,000 reconstructions with 500 million synapses, so what happens? It takes multiple steps. One is there's a type of a coloring step where you essentially, in a given section, try to isolate things which are evidently different from each other. And then there's a kind of section to section matching set where you try to find what's connected, what's matching. Now, and so they have all the tools that do this, and you're able to produce these are now remarkable structures. You get really, really deep neuronal structure through this process, right? And you get to understand their connectivity, synaptic connectivity now. We can really study how things differ at a synaptic level. I thought I had a 3D spinning thing here. Maybe we've had enough 3D spinning things for one day, but oh, there we go. There's a 3D spinning one. Yeah, so this is the kind of thing you can do. And there's a software package called Neuroglancer. It was developed in conjunction with a team, Viren Jain's group from Google. Google put a lot of resources into implementing the tools and techniques of these segmentations, and to allowing a browsing environment where you can say, using Neuroglancer, I can touch on this and I can go back and see parts of the 2D section where it came from, et cetera, things like that. Yeah. And anyway, you can go check this out. This is an implementation here from of all this data. It's publicly a data. There's projects you can get involved with from here. It's called Virtual Observatory of the Cortex. And there's the URL right there. So this is it's a really cool way to dive deeply into the EM data. Again, it's important to understand that without real advances in machine learning, this kind of segmentation would not be possible. And even with those, it requires still a proofreading, okay? You cannot miss, there's a huge burden on data quality because if too many sections are missed, you can't follow it at all. It can't be projected by. And even once the AI algorithms run, to do it carefully, you need a good proofreading kind of step. So, okay, so that's that. Well, we're kind of at time. Are we at time?
Yes.
Should we stop?
Like here and
Yeah, let's, here's what I want to do. Before the last thing I want to do is, I want to take a vote for people so we can, I wish I had, my wife was trying to get me to use these AHA slides. Do you know about AHA slides? It's a way to have, you can put people can put questions, you can use your phone, you can talk. And the trouble is that I looked at AHA slides and it would handle presentations up to 100 megabytes. And I'm like, one of my slides is 100 megabytes. So never did. But anyway, we can do our manual version of AHA slides. So I want to give you some topics and I want people to respond what they really want to hear about because we have way too much material here. This could be a whole course. So one is these, I'm just going to blast through some, well, I'm going to give you a flavor. We're going to do some teasers, okay? One is this is network approaches to analyzing data. This is like various kind of clustering approaches to using networks. It also includes some of the work of Lucas, some of the work on Morse, the basic Morse theory work. There's that. Another topic, the next topic in section 3, so that's one topic, network approaches. Don't think exclusively neural network approaches. It's some of that, but it's also other just network analysis. Okay. The next option is this is a deep dive into the problems and techniques of molecular cell type analysis. Okay. So this is more like transcriptomic analysis. The next section would be on cross-modality, multimodal comparisons. And the last section topics would be, this might be interesting to people. This is about integrating data across types. It uses different techniques of like variational inference. I think this is a little bit more mathematical. And so the variational techniques, I think I would like to include this one. I think this is a good one to include. So let's assume that we're going to do this one. And the last one is on cell type mapping. My feeling the best thing for people would be a deeper dive on the transcriptomic analysis and this variational stuff. I think that would be my preference, but people can vote and send in from the studio audience at home, send in your votes and we'll work it into the program. So, okay.
Sounds great. Yeah. Okay. Thank you. Let's thank Mike. The one with multi mapping also includes most open problems.
Thank you, everybody, for joining. We'll see you tomorrow at the same time. Thank you.